Bismillah ar-Rahman ar-Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about the process of the inflammation. Okay. In the previous video lecture, if you guys remember, we talked the inflammation introduction and we talked the disease which are related to the inflammation. If you missed that video lecture, so it is present into my channel. Okay. So before going to explain the process of the inflammation, here I am recommending to you that first of all you must watch the first lecture of the inflammation. Okay, so let's start the process of inflammation. Means that once the inflammation are happen, then what actually happening inside our body, what are the molecular mechanism of the inflammations. Okay, all these are actually mentioned in the process of the inflammation. So before going to explain the inflammation phenomena or the inflammation process or this medical condition, let me draw the blood vessels, let me draw the tissue, and let me draw all those amino cells which are usually involved in the inflammation. So let me draw each of the diagram. So first of all, let me draw the blood vessels. Okay, now what actually happen inside the blood vessel, there are the red blood cells, white blood cells, platelets, every blood cells are present inside these vessels. So let me draw the red blood cells. Now these are what? These are the red blood cells. They also contain WBC, but here I am drawing the neutrophils. Okay, although they contain monocytes, eosinophils, everything, but as we are explaining the inflammation, so here I will draw the neutrophil. So let me draw the neutrophil with this blue marker. They also contain neutrophils, and if you remember, we talked about the neutrophil in a detail. Okay, they contain multi-lobe nucleus. So let me draw the multi-lobe nucleus here. So let's say this is the nucleus of the neutrophils. Now neutrophil contain different kinds of molecules on their surface. So let me draw each of the molecules. They contain carbohydrates. So let me draw. And they also contain another molecule which we call the antigreens. So let me draw with the black marker. These are representing the antigreens. Antigreens are actually the adhesion molecules which are specially responsible for the attachment present on the surface of the neutrophils. So I will draw more neutrophil cells later, but not now. Now what actually after that, let's say that specific area are tissue. So let me draw here the tissue also. So let's say this is the tissue area. Okay, now what actually happen inside the tissue, let's say this is the skin area, let's suppose. Now inside the skin or inside the tissue, if you guys remember, we talked in the immunology lecture, we talked that tissue always contain dendritic cells and the macrophages. So let me draw the dendritic and macrophages here. Say this is the dendritic cells. Moreover, inside the tissue they also contain mast cells. So let me also draw the mast cell as well. So let's say this is the mast cells. Okay, mast, and they contain a small vesicles, or we can say granules, different kinds of granules, different kinds of the vesicles. Now these are the cells which are usually present in the skin tissue area. Okay, so what actually happened? Let's start the process. What actually happened? Let's say this skin area or this specific area of a body are become injured, or there is a wound. it become injured through a splinter, it become injured through a burn or any kind of the things, it become injured, the skin are become injured. So let's say here I am drawing the splinter. So let's say this skin area are become injured with the splinter. So let me draw the splinter here, which is very sharp. Okay, splinter are actually, it is a small fragments of the wood which is very sharp, and they can penetrate our skin as well. So let's say our skin is injured by this splinter. Now once, let me write, this is splinter. Now once the splinter are injected, due to this way, this splinter can also inject the microorganisms, because this splinter contain foreign particles, we can say microorganisms. So on the splinter they contain microorganisms. So let me draw. So let's say these are the microbes, bacteria, virus, fungi, euglena, any kind of the microorganisms it contain. Okay, now after that, what actually happen? Due to this rupture, or due to this penetration, the cells which are present at the skin area are become damaged, and due to this damaged, each of the cell which are damaged release different kinds of the chemicals. Some of them chemicals can activate this macrophages and dendritic cell. Macrophages and dendritic cell are present in the tissue, but they are not in the active form. The activation is maybe possible when the injured cell or the damaged cell produce the signals, chemical kind of the signals. Now each signals are for on the macrophage and dendritic cell. Let's say the macrophage and dendritic cells are activated. Once it is activated, each of the cells are reached to that specific area, and they cause the engulfing of each microorganism. So each microorganisms are now engulfed by the cells. Now after that, what actually happen? Once it is engulfed, so then it is break down into small fragments, and after that, if you remember, we talked that each cells are what? Each cells are the antigen presenting cell. Then they can present each antigen, small fragment of the antigen on their surface, means at that specific area. They can represent each of the antigen on MHC2 molecules. So let's say this is what these are the small fragments of the antigen. Now after that, what actually happen? This mast cell and dendritic cell will also release some kinds of the chemical which we call it chemokines. So they also release chemicals. So let's say these dots are representing the chemicals which we called it chemokines. The chemokines are injected, or the chemokines are internalized through this endothelial vessels of the blood. Once it is internalized, so what actually happen? They will activate the neutrophils. So the neutrophils are activated. If you remember, chemokines are those chemicals which can call upon the other cells. So usually here in the inflammation, they call upon the neutrophils. Now neutrophils are activated and they are attracted to that specific area. But how? Now, as I told you that neutrophil contain carbohydrates and antigreens. Okay, so let me draw. Let me write that this is what this is. Let's suppose antigreen. And these are what? These are the carbohydrates. Okay, now once the chemokines are internalized, due to this way, on the surface of each endothelial cells of the blood vessel, they contain selectin, and due to this chemokine, the selectins are expressed. So let me draw the selectin as well. Now once the selectin are expressed, what actually happen? First of all, the neutrophils are flow in the blood vessel at a high speed, but when the injury are happen, the speed of the neutrophils are become slow. Due to this way, not only the speed of the neutrophil, the blood cells, red blood cells speed are also become slow. Okay, which we call it constriction. Okay, so due to this accident, or due to the splinter injection, a contraction of the blood vessels are occur, due to which the vessels are become constrict, and the blood flow in the vessels are slow. And moreover, the neutrophils means the neutrophil movement are also slow. Now after that, what actually happen? The carbohydrate which are present on the neutrophil surface are just touches each selectin. Now once they touches each selectin, then what actually happen? These neutrophils can cause rolling motion. These neutrophils are present, but they can cause rolling motion. They are now rolling on each selectins. By this carbohydrate, the carbohydrate are attaches means touches the selectin, and due to this way these neutrophils are now rolling and after some times at that specific area, at that endothelial cells, there are some other adhesion molecules which we call it ICAM. Okay, so let me draw each of the, so let's say these are what, these are the ICAM molecules. Some of them are called ICAM1, VCAM1. Okay, so these are the ICAM molecule, which are the adhesion molecule. Now once it is rolling, so after some times the adhesion molecules of the neutrophil, which we call that integrins, and the adhesion molecules of these endothelial cells are both are bound to each other, due to which a tight junction are happen. So let's suppose if this is again the neutrophils, so let's suppose if this is again the neutrophils. Okay, so the neutrophils are bound here. Now neutrophils are bound to that area, and that the adhesion molecule of this neutrophils, which we call the integrin, and the adhesion molecule of the endothelial cell, both are bound to each other, and the tight junction between them are happen. Due to this way, let me draw the nucleus here. So this is the multi-lobe nucleus. Now due to this way, this neutrophils once attaches, then after that what actually happen? This damaged cells will also produce another chemical signals, another chemicals or molecules which we call 9 amino acid proteins molecules, which we usually call it bradykinine. So they also secrete another chemicals which we usually call it bradykinines. Okay, once it is fall on that area, they can cause they can increase the permeability of each endothelial cells. Means the space between each of the endothelial cells are become wide. So here are the space, it becomes more wider. So the space at that between the endothelial cells are become more larger, or the junction are become more larger. Now after that what actually happen? The neutrophil, when the neutrophil are squeezed at that specific area, now this is the neutrophil, they are squeezing from this junction area. This neutrophil have the ability, this neutrophil have that kind of property which we call the diapetasis. Once it is passes from that area and reach to this tissue, we call that, when the neutrophil are passing, we call that property diapetasis, and the process are known as extravasation. So let me write extra. This property is known as, whenever they are squeezing, we call that diapetasis. Okay, now once it is squeezing and reach to that area where the infection or where the problems are happen, so then the same function are also performed by the neutrophil, which the macrophages and dendritic cells do previous. Okay, as the macrophage and dendritic cells also doing the phagocytosis, similarly the neutrophils reach at that area, they are also start the phagocytosis process. Now after that what actually happen? These bradykinines will sometimes fall on that area, and sometimes this bradykinin will fall on the mast cell. Once it is fall on the mast cell, so they can activate the mast cell as well. Now once the mast cell is activated, they can fuse their granules. Okay, they can fuse their granules, and inside each granule they contain histamine and serotonin. So they release histamine and serotonin. Now histamine and serotonin, what they can do, this histamine and serotonin, so the space between each of the endothelial cells are become larger. Due to this way, the fluid which are present inside the vessels, let's suppose let me draw this, let's say these are the fluid. Okay, so the fluid which are present in the blood vessels are now passes through this junction and reach to this infected area, where the infection are happen, where the microorganisms are present. So what actually happen at that specific area, here the neutrophils are present, macrophages, dendritic cells, and each of the fluid. Okay, due to which the pus formation are happen, and we can say that due to this way the edema are happen. Okay, so the first thing in this case, the edema are happen, and whenever the neutrophils are present at that area, they can cause heating. What they can cause heat. A lot of neutrophils are present, due to which the heatings happen. Now moreover, due to this way, the damage area of the means this damage area are become healing, but due to this way the blood cells are reached to that area, red blood cells are reached to that area. At that red blood cell, it contain the thrombocytes. Now the thrombocytes are also reached to that specific area. Once the thrombocytes and the red blood cell are reached to that area, it can cause redness. What it can cause? It can cause redness. After that, what actually happen? More bradykinine are also released. So they can release more bradykinine. Now this time this bradykinine are fall on the capillary vessels. Once it is attached to the capillaries, then this capillary will produce prostaglands. What actually they produce? After that they will produce prostagland. Now what actually this prostagland do? This prostagland will stimulate the nerve ending. So they will stimulate the nerve ending. Now what does it mean? It means that pain is created. Okay, so how the pain is created? The pain are usually created in this way. Bradykinin are produced, they are fall on the capillary. Capillary can produce prostagland, and prostaglands will cause the stimulation of the nerve ending, due to which the pain is activated. Okay, so what we discussed the edema, which is usually activated when the fluid and the neutrophils and all of the cells are present at that specific area. We discussed the heat, how the heat is created during inflammation. Heat is created when all of the cells are fighting with the bacteria or the microorganisms. Then each microorganism and bacteria become die, due to which the pus or form, and it will create the heat. How the redness is created in the inflammation? The redness is created when this red blood cell are passes to that specific area and cause the healing of that wound, due to which the thrombocytes are also present at that specific area, and they can cause the blood clot, and due to this way the redness are happening at that area. Okay, now how the pain is created at that specific area? The pain is created at that specific area that this bradykinin are fall on the capillary, and once it is fall on the capillary, then the capillary will produce the prostaglands. Prostagland will cause the stimulation of the nerve ending, due to which the pain is created. Moreover, due to this way, the function of that specific area are also lost. Sometimes it is lost, or sometimes the function are not properly. Okay, a little bit the function are happen at that specific area. And how this function is lost? Because the nerves which is present at that specific area are become damaged. So that's why the function is also lost. But after some time the function is again gain when it is become recover or healed. Okay, so that's all about the inflammation. Okay, so after some times this area are become healed, and this area are become treat, we can say. So that's all about the process of the inflammation. I hope you understand about the inflammation process, or I hope you understand the molecular mechanism of the inflammation, or the mechanism of the inflammation. If still you have any kind of questions in your mind, so you can write the question in comment. Inshallah, I will give you answer as soon as possible. Okay, inshallah, in the next video lecture we are going to talk about the types of the inflammation. We are also going to explain each of the inflammation types in a detail, means chronic and acute inflammation. So please stay tuned and continue to watch the video. Thank you so much.